Hi friends, welcome to my channel Plants and Birds. Friends, in last video we have discussed about the reproductive tract part 1. Especially in that we have discussed about the ovary, in which the ova formation takes place. And in this video we are going to discuss about the reproductive tract part 2, that is oviduct, in which the ova journey takes place to form the egg, and how the air cell is formed in the egg we will discuss in this video. Firstly, oviduct. The oviduct is a long zigzag tube. It will not a straight tube, it is a zigzag tube consisting of glandular and muscular parts. Oviduct extends from the ovary to the cloaca, where the last ending part is called cloaca. It has 5 distant parts: infundibulum, magnum, isthmus, uterus, vagina. Firstly, infundibulum, approximately 9 centimeter in length and funnel-like structure. Capture ova from ovary. Okay, it will capture when the follicle rupture and the release of ova takes place. It will capture that ova from the ovary. Sperms from males are temporarily stored here for fertilization. Fertilization takes place if sperms are present. Ova remains 15 minutes here. Okay, infundibulum in which the ova will remain 15 minutes. Only become active just after ovulation. Okay, infundibulum become active just after ovulation. Okay, next continuation, how egg fertilization takes place. Cock generally can mate 10 to 30 times a day with hen. Semen ejaculation is 1 ml for first mating and then reduced to 0.5 ml or less. Okay. 1 billion, 100 million to 5 billion sperms are ejaculated per mating. Sperms are stored in utero-vaginal junction. Fertile eggs can be produced for 4 weeks after removal of males. Okay, this to be maintained in our mind. Fertile eggs can be produced for 4 weeks after removal of males. Movement of sperm from this junction to infundibulum. 30 minutes in absence of ovulation, if the ovulation is not is there means then it will take 30 minutes to move the sperm from infundibulum, okay, from the water uterovaginal junction to infundibulum. But in the presence of ovulation, only 5 to 10 minutes after an ovulation, okay, 50 sperms penetrate outer layer of the ova. Okay, less than 30 leads to poor fertility, so that 50 sperms penetrate outer layer of ova. Few sperms move to germinal disc. Okay, we will discuss what is germinal disc in detail. Okay, first one sperm unit with female pronucleus to form zygote. And next, this is the difference between, we can see what difference we can and differentiate the infertile and fertile eggs easily. That is how is, see, this is the diagram you will learn about it. In an infertile egg, it is a unicellular ovum. Okay, infertile egg is unicellular and contains haploid number of chromosomes. That is single set of chromosomes will be present, called blastodisc, and it is circular in shape. Diameter is 3.5 and with vacuoles in it. That is, see, it is infertile egg in which a white color vacuoles can be seen, and it is a less, okay, 3.5 mm is the width of the infertile egg, the yolk. And next, when coming to, whereas in fertile egg, it is a multicellular structure having diploid number of chromosomes, that is double. Okay, see, there is single set here, double set of chromosomes will be present, called blastoderm. Okay, it is oval in shape, not circular. Okay, oval in shape, which average diameter is 4.5 mm with no vacuoles. Okay, see, this is circular in shape, called blastoderm, and it contains no ovules. There is a no white formation cannot be seen, and it is clearly we can differentiate. It is a larger than, blastoderm is larger than blastodisc. Okay, it is 4.5 mm, it is 3.5 mm. And next, magnum, largest part of oviduct. Okay, Magnum is the largest part of oviduct. Major portion of albumin secreted here. Chalaza also secreted here. All four layers of albumin produced and completed except outer thin white. Developing remains here for white developing. Okay, all four layers of albumin produced and completely except outer thin white developing. Okay, here over remains for 2 to 3 hours. Only one type of albumin secreted in Magnum, but addition of water, rotation of egg make layers. Isthmus, small length of 10 centimeter, less developed than Magnum. Less developed than over remains here for 75 minutes. Okay, over remains here for 75 minutes. Inner and outer shell membrane secreted here. Outer membrane is about 3 times thicker than inner. Outer membrane is 3 times thicker than inner. Eggs from young birds have thicker shell membrane than the old birds. This must be remembered here. Okay, why some eggs are if it is also fertile, but why it is not giving birth? Okay, why it is not incubation? Why it is not giving birth? New chick is not developing. This is the reason. Okay, egg from young birds have thicker shell membrane than from old birds. Uterus, it is called shell gland. Okay, because here shell formation takes place. See, 10 to 12 centimeter long in laying birds. Okay, egg remains here for 18 to 20 hours. It will be remain here only. Fourth layer of albumin is completed here by addition of water and salts. Okay, albumin, what is albumin? Now we will study about. Consisting of four layers: chalaziferous or inner thick white, which forms 3%, inner thin albumin 17%, outer thin firm or dense albumin 57%. This is important. Outer thin firm is 57%, okay, maximum percentage. Outer thin albumin 23% of total albumin. Okay, the calciferous layer very close to the yolk, immediately surrounding the vitelline layer of the yolk. This layer twists into two cords on either sides of the yolk called chalaza, which form due to rotational movement of the egg in the oviduct. Okay, these are the parts of the egg. Okay, this is chalaza which is formed due to the rotation of the egg in the oviduct. Okay, nucleus, blastoderm. Okay, this is the fertilized egg. It's called blastoderm. Fibrous layer. Okay, chalaziferous layer we can easily see. Vitelline membrane. Okay, vertebra is the middle portion of the yolk, yellow yolk, white yolk. You can see here. Outer thin albumin. Okay, it is a main portion, and inner thin albumin. Fibrous layer will be present in between. And next, continuation. Chalaza hold the yolk central position and thus serves as an anchor for yolk. How to move? It will be anchored by the chalaza. Chalaza contains a protein called lysozyme, which is possessing antimicrobial properties. Helps to prevent the microbial spoilage of the egg. Okay, what prevents the egg from microbial spoilage? As it is an organic food, why it is not spoiling means it is a main reason. Okay, it contains a protein called lysozyme, which possesses antimicrobial properties. Aging, improper storage, and microbial spoilage make thick albumin watery. Okay, thick albumin become watery. Okay, aging, improper storage, and microbial spoilage. If any microbes, okay, dangerous microbes enter, means then it will become thick albumin, become watery. Next, approximately 15 ml fluid is added by plumping process. Okay, plumping process is a process where fluid is added in the eggshell. It pigment and cuticle is added here. Pigment and cuticle is added here in eggshell. Okay, as I told, shell gland. Okay, 2 to 2.5 gram calcium is used for shell formation, for which 53% taken from the feed, 47 from the bones. So that every poultry grower, okay, or for purpose of egg growing, they must give the sufficient calcium. Otherwise, the strength of the bones may be decreased in many of the ends. Okay, so that, see, however we can give 53% only taken from the feed, 47 from the bones only, so that 2 to 2.5 gram calcium is required for cell formation for a single egg. Next, vagina, last part of oviduct and 12 centimeter long. Egg remain off for few minutes here. No egg formation occur here, only expel the egg outside the body. Sperm stored glands are present at junction of shell gland and vagina. It can store sperms for 10 to 14 days. Okay, this is about the oviduct. Thank you, friends. Subscribe if like my video and subscribe to get more information about the poultry farming and other agriculture related topics. Thank you, friends. Bye.